Hi guys, and welcome to our first lesson in the endocrine system. We're going to start this lesson by reviewing some things that we should already know about the endocrine system and how cells communicate with one another. When it comes to the human body, our cells have four principal mechanisms in which they can communicate. The first is called gap junctions. Gap junctions are specialized membrane structures that allow cells to communicate by exchanging ions, nutrients, and other small molecules. But these holes or pores within the cell membrane will connect one cell to another so that substance can move from one cell and into the other. The second is neurotransmitters. Neurotransmitters are chemicals that are released from neurons, and they'll travel across a space called a synaptic cleft, and they'll arrive at a secondary cell. That secondary cell can be another neuron, or it can be a muscle or a gland, and that neurotransmitter will cause some kind of effect in that secondary cell. The third type is called paracrine. Paracrine is when substances will be secreted from a cell into the extracellular fluid that's surrounding it. And that substance will diffuse through that fluid and arrive at neighboring cells. It's very similar to hormone secretion. However, it's going to stay localized and it's only going to have an effect on these neighboring cells. Hormones will travel through the blood and they'll have an effect on distant tissues and organs rather than just their neighboring cells. So gap junctions, the structure of a gap junction, they are made up of clusters of channels that form when two neighboring cells' membranes come very closely together. And they're going, so you can see in this animation, there is the two neighboring cell membranes and these clusters. Well, they're going to function to allow the direct transfer of ions and small molecules between these two cells. This plays a crucial role for many cellular processes such as growth, cell differentiation, and homeostasis. With neurotransmitters, we're going to see an action potential changing the voltage of the cell membrane until it arrives at the end of an axon terminal called the synaptic end bulb. Inside of that synaptic end bulb, there are vesicles, and those vesicles contain chemicals that are neurotransmitters. Those neurotransmitters will be released from those synaptic vesicles, and they'll have to travel across that synaptic cleft. And you'll see that secondary cell has these little orange projections sticking off of it. Those are receptors for that neurotransmitter. You can see this animation here. There's the neurotransmitter being released and binding to that receptor, and then we will have an effect in that secondary cell. If it's a gland, it will release a hormone. If it is a muscle, there will be a contraction stimulated. If it is a neuron, there will be a second action potential continuing that signal. with paracrine signaling, we're only going to have an effect on the neighboring cells. So we're going to have a cell that will produce a substance, a chemical, and that cell is called the secreting cell because it makes a product and it sends it out. The target cells of this secreting cell are going to be right next to it, so it's only going to affect the neighboring cells. And then we have hormones. So when a cell of the endocrine system releases a hormone into the bloodstream, it could be a thyroid cell, an adrenal gland, a pituitary cell, just any cell that produces a hormone, and that hormone is secreted into the bloodstream. It will travel through the bloodstream until it reaches what is called its target cell. What the target cell is depends on what hormone we are talking about. So we need to review this whole concept of glands. So what is glandular epithelium? Well, a gland is made up of cells that secrete a fluid substance that is needed by the body. The cells that secrete these fluids are specialized to secrete materials. It could be a digestive juice in the stomach. They could be hormones. It could be milk production in the mammary glands, or sweat production in the skin, or even wax production in your ears. Well, glands are going to be classified in two different ways. They're classified on their complexity, but they're also, and what we're going to focus more on is how they're classified with the way they release their substances. So the two types of glands are exocrine glands and endocrine glands. So exocrine glands always secrete down a duct onto an epithelial surface, and it can secrete multiple different substances. Where an endocrine gland, those are ductless. There are no ducts, and they only secrete their substances into the bloodstream. So an example of an exocrine gland would be a sebaceous gland that's found in the skin, while an example of an endocrine gland would be the thyroid. Our endocrine glands are going to have specific characteristics. These glands secrete hormones directly into the blood. That way they can travel longer distances. Endocrine glands are also going to be very, very tightly packed together, and there's going to be a capillary bed very closely associated and running through that glandular tissue. That way, the cells can release their substances and they can diffuse and get into that bloodstream. Cells of the endocrine system will release their substances through exocytosis into the bloodstream. So for example, we have the thyroid, the thymus, the adrenal glands, and the pancreas. And so this is a picture that you can see the difference between that exocrine and endocrine gland. The picture on the left, the exocrine gland, you can see a layer of epithelial cells. And that substance, in this case it is sweat, it will be released directly into a duct, and then that duct will end up going to the surface of the skin to release the sweat. Versus the endocrine gland, those cells are going to line blood vessels. So that way, when they release their substance, it goes directly into the bloodstream. These are some histology images of a sebaceous gland. You can see if you look at the picture over to the left, there are layers of the skin. You have the most superficial layer of the epidermis, and you can see the middle layer of the dermis that has the main excretory duct going through it. And down in the hypodermis, you can see that sebaceous gland. If you look at the image in the middle, you can see those that secretory region of the sebaceous gland a little bit better. You can see those cells, and you can see the neck of that sebaceous gland and the opening to the surface. The image over to the right is showing a more clear image with the hair follicles still in place, but you can see that secretory region of the sebaceous gland with those simple columnar epithelial cells, and you can see that neck of the sebaceous gland leading to that opening. What you're looking at here is a histology image of the thyroid gland. So when you see these roundish pink structures, these are called follicles. And those follicles are actually storing a thyroid colloid, which is the storage form of the thyroid hormone. But if you look around these follicles, you'll see a simple cuboidal epithelium. You can see those round nuclei, and that's what's going to form the walls of these follicles. And if you look at the spaces in between the follicles, you're going to notice capillaries that are lined with a flattened dark stain nuclei. So for example, right where I have my laser pointer is a capillary, and then you can see that epithelium is kind of squished and the nuclei are that darker purple color. The endocrine and the nervous system work very closely together. They have some similarities, and of course they have some differences. So some differences are, if you think about the nervous system is composed of the brain, the spinal cord, and nerves, and everything's connected, so it's wired. Versus the endocrine system will secrete that hormone into the blood, and then it gets carried through the body. So it's kind of more of a wireless system. The endocrine system, because it relies on being carried through the blood, it's going to take longer for the hormone to reach its target site. So it's going to be more slow acting, taking seconds to possibly even days, versus the nervous system is very rapid. Our nervous system communicates within less than a second. We're talking milliseconds. The nervous system, though, it's typically going to be shorter acting as well. It will stop faster. And it's going to have a more targeted location. It's going to be more one target area or organ, versus the endocrine system will last longer because that hormone will be transporting through the blood, and there are going to be very specific receptors that bind and interact with that hormone. So any cell or organ that has that receptor will respond to that hormone. So the effects can be more widespread throughout the body. The endocrine system is also going to be a little slower to adapt to some long-term stimulus versus the nervous system is much more quickly adapting. The nervous system and the endocrine system work together to help maintain homeostasis in the body, and they have some similarities. These two systems can actually regulate each other. The neurotransmitters that are released from the nervous system will have an effect on glands and the hormones that they secrete. And then in turn, those hormones can have an effect on neurons. Many chemicals within the nervous system and the endocrine system function as both hormones and neurotransmitters. So for example, dopamine and epinephrine or norepinephrine are both hormones and neurotransmitters found in both systems. They can also have similar effects on their target cells. There are also a not as talked about specialized cell called a neuroendocrine cell. And these are actually neurons that can secrete hormones. And a very nice comparison of these two systems is found in the first chapter of your book, table 17.1. So this image is showing you a side-by-side comparison of how the nervous system can relay a message versus the endocrine system. So what you're seeing is a nerve impulse will be detected by these dendrites. That signal will get carried towards the cell body and then down the axon of that neuron. It will continue to go down to these axon terminals where eventually it will meet a synaptic end bulb. And in that synaptic end bulb, there will be a neurotransmitter that gets released to have some sort of effect on those target cells. Versus the endocrine system, we have these endocrine gland cells that are producing and secreting their substance, their hormone, and that will get secreted into the blood. That hormone's then carried through the blood and it will diffuse into the surrounding area of those target cells. So hormones are produced by cells that are specialized to make that hormone. Cells will secrete their hormone into the extracellular fluid that is surrounding the cells. And then that hormone will diffuse into a capillary bed and enter the bloodstream. Blood will then transfer hormones to their targeted cells. So that hormone will then diffuse and bind to a receptor on that cell. And then that hormone binding to the receptor will regulate the activities of that cell. So once a hormone reaches that targeted cell, what we're going to find is on the cell surface is a receptor. So if you recall way back from biology, the cell membrane is a phospholipid bilayer, meaning there are two layers of these phospholipids. The outer layer, the head of that phospholipid, is very hydrophilic. So it really likes water. So it loves that watery environment on the outside of the cell. And then these hydrophobic tails are water fearing. So they're going to face each other. And on the other side, we're going to have those hydrophilic heads that will be next to the watery internal cellular environment. So if we look at the external cell membrane, the outside of the cell, there's going to be what's called a receptor. And that receptor is going to have a very specific shape. So then when the hormone diffuses out of the blood and into that space around the cell, that hormone will bind with that receptor and it'll fit right into it, kind of like a puzzle piece. So if there is no receptor on the cell, then there will be no effect in the cell. That cell that does not have a receptor is not a targeted cell. Only cells that have the receptor will be able to have the hormone bind and then respond to that hormone. So these targets can be organs or specific cells within a tissue. But these cells have to have a receptor for the hormone and can respond to it. Some target cells may have an enzyme that will actually convert the hormone to its more active form. So the endocrine system is made up of glands that secrete substances into the bloodstream. So we're not talking about any exocrine glands that use ducts. And it's going to function similar to the nervous system for communication within the body and control of body functions. But it's going to work a little bit slower than the nervous system does. So these hormones are chemicals that are released from one part of the body, and they're going to be carried through the bloodstream to affect another part of the body. These hormones that are released from the cells are going to be released in very small quantities, but they're very, very specific. So the target cells have to have a receptor for that specific hormone. And if the hormone contacts the cells without these target cells, there is not going to be any effect because they don't have the receptor. And there are two major types of hormones. We have water-soluble hormones that are made up from amino acids. And then we have lipid-soluble hormones that are made up from steroids. When it comes to the steroid-based hormones, these are lipid-soluble hormones. So what that means is they can pass directly through the membrane. Because remember that cell membrane is made up of lipids and like dissolves like. So this lipid-soluble hormone will pass directly through the cell membrane where it will find a receptor on the inside of the cell. That can be within the cytoplasm or it could be within the nucleus of the cell as well. But when it does find its receptor, it's going to form a hormone receptor complex. And that will then alter what the cell is doing. So it will eventually turn on a gene. Remember that a gene is a section of DNA that codes for a protein. So this complex can either activate sections of that DNA or deactivate sections of that DNA. And so we're going to have some kind of cellular response. This is going to include our sex steroids, our corticosteroids, and calcitriol. There is an exception of the thyroid hormone. The thyroid hormone is a monoamine, which we're going to talk about on the next slide. However, it is hydrophobic, so it will function like a lipid-soluble steroid hormone. Our non-steroid hormones, these are the monoamine hormones. And so they are water soluble and amino acid based. So what that means is that cell membrane is not going to let them pass through, because in the inside of that cell membrane is that hydrophobic portion, those hydrophobic tails that really do not like to mix with water. Water is polar. That charge isn't going to be able to pass through that cell membrane. So there is going to be a receptor on the outside of the cell that's very specific to that hormone. And that hormone binding to the receptor, that is the first messenger. And non-steroid hormones require the use of a secondary messenger in order to continue the message. It's kind of like running a relay race, and you have to pass the baton to the next runner in order to finish the race. So when that hormone binds to the receptor, that is going to activate this secondary messenger, and it will mediate the response within the target cell. So these are going to include things like proteins, peptides, and those monoamines, with the exception of the thyroid hormone. So the two main categories of hormones are the non-steroid hormones and the steroid hormones. Non-steroid hormones are going to be water soluble, and there are three subcategories. We have monoamines, which include catecholamines, melatonin, and the thyroid hormone, where peptides include insulin, antidiuretic hormone, oxytocin, and gonadotrophin, and proteins. Proteins include erythropoietin, which stimulate red blood cell production, follicle stimulating hormone, luteinizing hormone, and thyroid stimulating hormone. The steroid hormones, those include things like progesterone, estradiol, cortisol, testosterone, and aldosterone. If we take a look at how these steroid hormones are produced, in this image, you can see the center molecule here. This is a molecule of cholesterol, and that is the precursor for these steroid hormones. This cholesterol will get converted into a precursor, which will then get further converted into different hormones based off of this cholesterol molecule. So this molecule can be converted into the aldosterone, or it can be converted into progesterone, or it can be converted into the testosterone. When it comes to protein hormone synthesis, this is going to be where I really encourage you to go back to what you learned in biology about protein synthesis and transcription and translation, because protein hormone synthesis is going to be very, very similar. We're going to have a section of the DNA that codes for a protein be activated, and that DNA is in the nucleus and it can't get out. So RNA polymerase is going to come in and make a messenger RNA molecule that can leave the nucleus. That messenger RNA will find a ribosome where the ribosome begins to read the messenger RNA. As it reads that messenger RNA, it's going to use transfer RNA molecules to go out and bring in the correct amino acid. And the ribosome will connect those amino acids together and start to form a polypeptide. So as the amino acids chain starts to form, this chain isn't a hormone yet. It's more of a pre-pro hormone. So once that mRNA molecule leaves the nucleus, it's going to find a ribosome. And in protein hormone synthesis, that ribosome is actually going to be on the endoplasmic reticulum. So as the ribosome on the endoplasmic reticulum starts to build that pre-pro hormone, there are going to be enzymes inside of the endoplasmic reticulum that start to chop off sequences and begin to break it up. Once that happens, now it is considered to be a pro-hormone, and it will move out of the endoplasmic reticulum to the Golgi apparatus by a vesicle. In the Golgi apparatus, it will continue to get further modified, and once it's done being modified in the Golgi apparatus, it will be put back into another vesicle, and it will begin to move towards the cell membrane so it can be released. Well, while it's in that vesicle, there are enzymes that will continue to modify it. So that way, once it reaches the cell membrane, the hormone is now in its active form instead of a pro-hormone. And then that hormone can diffuse into the bloodstream and be carried to its target location. Over here on the right, this is a molecule of proinsulin that would be packaged as a pro-hormone into one of these vesicles. And you can see that this blue section has been removed. It's been cut and modified. And so I then have the active, the red portion, it's been removed from the green portion, and I now have my active form of insulin. When it comes to monoamine hormone synthesis, almost all of these hormones are going to come from the amino acid tyrosine. So there's one exception, and that's in the case of serotonin and melatonin. Those will be synthesized from tryptophan. So you can see here I have a molecule of tryptophan, and there'll be a series of steps that are performed by enzymes converting that amino acid into melatonin. And we know that they are enzymes because if you look at the end, they all end in ACE. And anytime you see ACE, we know we are working with an enzyme. When it comes to the catecholamines, these are your fight or flight response. So it includes epinephrine, norepinephrine, and dopamine. And this also will be performed by a series of steps, by enzymes, which all end in ACE, But in this case, we can see we have a molecule of tyrosine, and tyrosine is converted to dopa, and then dopa is converted into dopamine, and then dopamine will be converted into norepinephrine, and then there will be another enzyme that can convert norepinephrine into epinephrine. When it comes to how these cells get around the body, there are different ways they'll be transported depending on if they're hydrophobic or if they're hydrophilic. So if they're hydrophilic, meaning that they are easily dissolved in water, like the monoamines and the peptides and the proteins, those can be dissolved in the blood plasma because blood plasma is mostly water. So they'll mix with the blood plasma and be able to be transported. However, if they're hydrophobic, they're not going to want to mix with that water, and they're going to have to be transported by binding to carrier proteins like albumin and globulins that are produced by the liver. When the hormone is bound, it will have a longer half-life. When the hormone is unbound, then it is not protected from being broken down by enzymes and being filtered by the kidneys. So it will have a much shorter half-life. But only the unbound hormones will be able to leave the capillaries in order to reach those target cells. But the proteins that are being carried by those the hormones that are being carried by the transport proteins, those are going to be protected because they won't be able to be broken down by liver enzymes and kidney filtration. So hydrophobic hormones that are transported this way include those steroid hormones and the thyroid hormone. So as we go through the different glands and hormones, you'll learn more specifics about the effects of these hormones, but these are just some general effects that are produced in these target cells. The first is that they can change the plasma membrane permeability and allow some substances in, or maybe prevent substances from entering the cell, or it could change the potential or the charge of the cell. It could stimulate protein synthesis, transcription and translation. It could also activate or deactivate enzymes. Hormones can also cause cells to secrete substances, like in your stomach, the secretion of hydrochloric acid or the secretion of insulin in the pancreas. Or they could also stimulate mitosis, which is cellular division that's supposed to replace old and damaged cells. Hormones will only stimulate those cells that have receptors for that specific hormone. If the cell does not have a receptor, it will not respond to that hormone. Receptors can be in two locations. If it is a water-soluble hormone, that receptor can be a protein or a glycoprotein. That's a protein with some sugars on it. That's what a glycoprotein is. Those will be found on the surface of the cell. Or if it is a lipid soluble hormone, then it will be found on the inside of the cell within the cytoplasm or the nucleus. And receptors are specific for each hormone. So that is called specificity. So how genes are activated are in two different ways depending on the type of hormone. Those water-soluble hormones that cannot get into the cell are going to rely on what's called the second messenger system, where the lipid-soluble hormones that can diffuse past that cell membrane, they will use direct gene activation. So in direct gene activation, this is where that hormone can diffuse through the plasma membrane of its target cell. And in this example, the hormone will diffuse past the membrane of the nucleus, and it will find its specific protein receptor within that nucleus. The hormone binding to the receptor will make a hormone receptor complex. And that hormone receptor complex will bind to a specific site on the molecule of DNA. And that's going to activate that specific section of DNA. Remember that a gene is a section of DNA. So that gene will be activated, and the result will be the synthesis or production of new proteins. So then we'll have transcription occur in the nucleus, and translation will occur in the cytoplasm on a ribosome. When it comes to our non-steroidal hormones, these are the ones that cannot get into the cell membrane, so they are going to rely on a second messenger that is located inside of the cell. And we're going to have kind of a molecular relay occur here. So you're going to have a hormone that is your first messenger binding to a receptor on the surface of the cell. That hormone binding to the receptor is going to activate G protein. G protein is an enzyme that will activate adenylate cyclase inside of the cell. And adenylate cyclase will catalyze our adenosine triphosphate into cyclic AMP that we call CAMP. CAMP is the second messenger. CAMP will activate a protein kinase, and that protein kinase will have many different metabolic results. Depending on where we're at and which cell, it can activate enzymes or it can deactivate enzymes, but it will induce some sort of cellular response. And then once the achieved goal or desired goal is achieved, CAMP is going to have to be broken down, and it's broken down by an enzyme called phosphodiesterase. So in this image here, you can see if you locate the number 1, laser pointer, right here, that is the hormone as the first messenger, and as it binds to that receptor, it is going to activate G protein. G protein is then going to, in turn, activate adenylate cyclase. That is going to convert this molecule of ATP into cyclic AMP and diphosphate. Remember, adenosine triphosphate has 3 phosphate molecules, and over here where we have the 2 phosphates and cyclic AMP. So cyclic AMP will activate an enzyme called protein kinase. And then that protein kinase can activate and deactivate proteins and have various metabolic effects. Wide range of possible effects on the cell. And that brings us to this concept of amplification. So we have a very small amount of hormone that is produced by the gland, but in the end, we're going to result with a very large effect because every step of the way, the result is being amplified. So when we get to our final product, we have a big effect. And how sensitive these target cells are to hormones can be upregulated and they can be downregulated. They can be changed because our cells can change the number of receptors that they have on the surface to respond to this hormone. In upregulation, the number of receptors on the surface is going to be increased, and that in turn will increase your sensitivity to that hormone. Whereas downregulation is the opposite. This is where we're going to have a reduction in the number of receptors. So the cell can actually kind of pinch off part of its cell membrane that has these receptors in a process called endocytosis. And then it will internalize that part of the cell membrane. And that will reduce the number of receptors on the cell surface. And therefore sensitivity will decrease. And that can occur if we have exposure to hormones, high hormone concentrations for long term. Eventually, our hormone signals, though, have to be turned off because they've already done what they need to do. They've served their purpose. So hormones will be taken up and they'll be broken down by the liver and the kidneys. Those hormones that are broken down by the liver will be excreted in the bile and through the feces, whereas kidneys will filter and excrete those hormones in the urine. How fast hormones are removed from the blood is called the metabolic clearance rate. And when we talk about metabolic clearance rate, we often refer to the half-life, which is the time that's needed to clear half of the hormone from the blood. So the slower the metabolic clearance rate, the longer the half-life of the hormone will be, versus faster the metabolic clearance rate, the shorter the half-life will be. And so this was just the general hormone introduction. In our next lesson, we will go over the anatomy of each one of these glands. We'll go over the hormones each gland secretes and the specific effect that each one of these hormones have on the body. So we'll discuss the thymus, pancreas, adrenals, pineal, pituitary, and hypothalamus, as long as the testes, ovaries, the thyroid, and the parathyroid. Thank you for watching, and I look forward to seeing you guys tomorrow.